Question number 31. The diagram shows oxygen diffusing from the space inside an alveolus into the blood through the gaseous exchange surface. What would increase the rate of diffusion of oxygen from the alveolus to the blood? So definitely if concentration here increases, then the movement would be more faster, right? And decrease in this concentration, of course. Oxygen. Oh no, actually, we can do one more thing. We can so if the wall is thinner, rate of diffusion is faster. So rate of diffusion is faster. We can just make this decrease the thickness, and we can just keep this same, or even if that decreases, that also works. So first is to increase concentration of X. I can only see X in one option, and decrease Y, yes, and keep the same. Yes, W could be kept the same or maybe decrease, but that we don't need to consider that because there is no option suggesting anything that. Which tissues are present in a bronchus? So they do have cartilage, right? Cartilage they have. Then they also have smooth muscle. So this is again the theoretical knowledge. As I was talking in this question, we would look at the table. See, from that one table so many questions come. It's very important, that one table. So which tissues are present? So cartilage is present, so that is wrong. Ciliated epithelium, it is also present, right? So that is wrong. And smooth muscle, that is also present. So answer should simply be 32A. Question 33. Steep concentration gradients must be maintained for efficient gaseous exchange to occur in the human lungs. Which row correctly describes how steep concentration gradients can be maintained? Elastic fibers in the walls of the alveoli recoil. Yes, of course, recoiling will allow more force and more pressure. So that has to be in the options. Answer will be between A and C. Continual supply of deoxygenated blood. Yes, this would allow for a steep gradient to be maintained for ventilation. So answer should be A. Question 34. Scientists investigated the effect of an antibiotic on the treatment of cholera. 320 people with cholera were divided into two groups. One group was treated with antibiotic, while the other group was not given the antibiotic. Both groups were given fluids containing sugars, and mineral salts, which is oral rehydration. The saline we have. The scientists recorded a number of days that each person had diarrhea. What is the percentage decrease in the mean time that the person had diarrhea when they were treated with antibiotic? So decrease from this to this. So the decrease here is 2.1, and the initial was 5.3, and multiply that by 100. We should have 2.1 divide 5.3 into 100, which is, that gives you 39.6 as per the calculator. Answer is A. Okay. How does antibiotic penicillin affect the metabolism of a bacterial cell? So what it does is it prevents crosslinks between the cell walls. Crosslinks prevented. So that would be the answer, and it does not allow for more growth or metabolism. Reduces cell growth but prevent wrong. Inhibits the formation of crosslinks. This should be the answer. Penicillin inhibits the hydrolysis of peptidoglycan. Not actually hydrolysis. It rather increases, it rather prevents the crosslinks from forming. So it does not inhibit the hydrolysis. Rather, it allows the inhibits the formation. Penicillin weakens the cell wall. It does not weaken. It just doesn't just allow for the formation of the wall. Question 36. Scientists studied the multi-drug resistant bacteria infections in children caused by one type of bacteria between this to this. The percentage of multi-drug resistant infections rose from 0.2% to 1.5%. What was the percentage increase? So it increased by 1.3, and initial was this into 100. Divide 0.2 into 100. So the increase is a 650. Answer should be D. Whooping cough is a highly infectious disease of the gas exchange system caused by the bacteria. This one, Bordetella pertussis, which method provides protection to infants against whooping cough and reduces chance of developing this disease. A short-term course of more than one type of antibiotic. So this is actually after the disease occurs. This would be effective after disease occurs. A 6-month course of one type of antibiotic. Again, this would be after the disease occurs, so would be more effective. Injections of antibody specific, not really. Antibodies are not going to be permanent. Yes, antibodies. Antigens. Antigens would give permanent, right? Formation of memory cells. Like it will allow for the formation of memory cells, so future infections can be prevented. The electron micrograph shows a type of blood cell. What can we confirm in the electron micrograph? So we can see a lot of rough endoplasmic reticulums, means it does a lot of protein synthesis. Isn't that so? It looks like it does a lot of protein synthesis, hence that could be the answer. Well, the cell secretes products that are toxic to pathogens. We cannot deduce this from there. I cannot see many lysosomes or any hydrolytic enzyme source. The cell synthesizes, this could be the option actually. The cell synthesizes large quantities of antigens. I can't see that actually, and it cannot synthesize antigens actually. The cell digests pathogenic bacteria. I can't see. We cannot actually deduce that this is a phagocyte from this diagram. So I will actually go with answer B. Question 39. Why is mitosis important in the immune response? It allows B lymphocytes to produce plasma cells. We've got the answer on the very first option itself. It allows B lymphocytes to synthesize antibodies. Wrong. It allows neutrophils to carry out phagocytosis. Also wrong. It allows T lymphocytes to recognize foreign antigens. Wrong. It just allows for more cell division, right? And hence it allows for it does not allow for any type of synthesis or anything. It allows for more cell division simply, and that is allows for more plasma cells to be formed. What is fused with B lymphocyte to form a hybridoma cell in monoclonal antibody? So myeloma cells, which are the cancerous cells. So those are combined with this. So myeloma cells. This should be the answer. So answer is D. So guys, that is the paper, and I hope it was helpful. I tried to give the descriptions for every question. Some questions, of course, it's really tough to give any descriptions because those are mainly theoretical knowledge, and you just need to know the concepts from the textbook or any other resource you use online or stuff. So yes, I will be bringing more biology papers for sure. Next paper could be maybe a pure math or a mechanics paper, maybe a physics as well. So I hope that was helpful, and as I always say, stay tuned and peace.